somewhere between the surface and about 650 feet down, in water that is still blue and still lit, there are animals swimming that you would not see if they were 18 inches from your face, not camouflaged, not tucked behind anything. There is nothing to tuck behind out there. No rock, no weed, no overhang, no seafloor within a mile in any direction. Just water, lit from above, and a body moving through it. And you would look straight past them. In the late 1990s, two biologists, Sonka Johnson and Edith Witter, decided to put an actual number on that. They collected 29 species of soft-bodied, jelly-type animals out of the northwestern Atlantic and the Gulf of Mexico, kept them alive, and measured how much light went through each body without being absorbed or knocked off course. Living tissue, on an optical bench, in the visible range that our own eyes work in. The results ran from about 91% for one small hydromedusa, down to about half of 1% for a swimming sea snail called Cleone limacina, the one people call the sea angel. 91%, that is a whole animal. Muscle, nerve, gut, gonad, the lot, and 9/10 of the light just carries on through as if the animal were not standing in its path. Now put that beside the land. Take a walk through any forest on the planet and try to find a transparent vertebrate. Try to find a transparent anything, really, above the size of an insect wing. Johnson went on to write a review of this whole field in 2001, and one of his flattest, least decorated conclusions was that transparency is essentially a pelagic trait. It is common in the open water column. It is uncommon in other aquatic habitats, in rivers, in tide pools, along reefs, and on land it is close to non-existent. That asymmetry has nothing to do with which animals happen to evolve where, and nothing to do with the ocean being older or stranger or more inventive. It comes down to a single number, and once you know the number, you can predict most of what follows. But before the number, the requirement. One fact needs to be nailed down at the start, because everything after it is downstream. There are exactly two ways for a body to be visible. Two. That is not a simplification for the sake of narration. It is the physics. The first is absorption. Light goes into your tissue and does not come out. Some molecule in there catches a photon and converts it into something that is not light anymore, usually heat or a chemical change. Melanin does this. Hemoglobin does this, which is why blood is red and not clear. Carotenoids do this. Chlorophyll does this. The pigments in your retina do this on purpose. Absorption is what makes a shape look dark. If you are floating in blue water and there is a sunlit surface above you, an absorbing body reads as a silhouette, a hole in the light. A predator looking up from below sees you the way you see a bird against an overcast sky. The second is scattering. Light goes into your tissue and comes out, but not in the direction it was traveling. It bounces. It bends. It gets deflected off course, and instead of continuing down toward the deep, some of it turns sideways and reaches an eye that was not supposed to receive it. Scattering is what makes a shape look bright, or milky, or cloudy. Not dark, cloudy. This is the reason a glass of water is clear and a glass of milk is white, even though milk is mostly water. This is the reason a solid block of ice is transparent and crushed ice is white. Same substance. Same chemistry. All you have done is add surfaces, and surfaces are where light changes direction. So the design brief for an invisible animal in open water is short enough to write on a napkin. Get rid of everything that absorbs. Get rid of everything that scatters. Do this without dying. That second clause is where it gets interesting, and it is where this whole thing eventually breaks, because two of the organs you would need to keep in order to survive are, for reasons built into physics rather than biology, extremely hard to stop absorbing and scattering. Not hard as in nobody has gotten around to it. Hard as in the function of the organ is the thing that makes it visible. An eye has to bend light. That is what an eye is for. A gut has to hold food. And food, before it is digested, is another animal's body, complete with all the pigment and structure that animals spent its life building. Every transparent animal in the ocean is, in the end, a set of engineering decisions arranged around those two problems. And once you have the two problems in your head, you can look at almost any transparent creature and read its history off its own body, where the compromise was made, which organ got sacrificed, which one got hidden behind a mirror, or shrunk, or pushed out on a stalk, or wrapped in the one color that is invisible in the place where it lives. That is the case I want to lay out. The requirement first, then the candidate solutions, then the two failures nobody has engineered around, then the workarounds that exist precisely because those failures are permanent. Start with the requirement, because the requirement is stranger than it sounds. Absorption is honestly the easy half. Pigment is a thing you make. If it costs you more than it earns, evolution can stop making it. There are whole lineages that have simply put down the paintbrush. The Antarctic ice fish has lost hemoglobin entirely and run on plasma alone, which works because cold seawater holds a lot of dissolved oxygen, and those fish live slowly. Leptocephali, the ribbon-shaped larvae of eels and their relatives, carry almost no hemoglobin during the larval stage, which is one reason you can hold one up and read a newspaper through it, assuming you can find one and assuming you still have a newspaper. Losing pigment is a normal evolutionary move. Cave animals do it. Deep-sea animals that never see light do it. It happens constantly. Scattering is the hard half. Nobody makes scattering. It happens on its own, automatically. Wherever the optical density of your body changes from one place to the next, every cell membrane, every mitochondrion, every fat droplet, every protein granule, every nucleus, every boundary between one tissue and the next. You are not built out of a substance. You are built out of billions of tiny compartments, and every wall between two compartments is a place where light can turn, which means an animal cannot achieve transparency by not doing something. It has to achieve it by rebuilding what it is made of. So, the number. Light does not travel at the same speed through everything. In a vacuum, it goes as fast as anything can go. Push it into glass or water, or the jelly inside one of your own cells, and it slows down. The ratio between those two speeds is called the refractive index, and it is the single most useful number in this entire story. Air, at sea level, has a refractive index of about 1.0003. Call it 1. Light barely notices air. Seawater sits at roughly 1.34. Light moves through seawater at about 3/4 of its top speed. And living tissue? The watery part of a cell, the cytosol, comes in around 1.35 to 1.37. The structured parts, the membranes and the protein-dense granules and the fat, run higher, somewhere in the region of 1.39 to 1.47, depending on what exactly you are measuring and who is measuring it. Look at those three numbers side by side, and the entire geography of transparency on this planet falls out of them. Tissue against seawater is a difference of maybe 1 or 2 hundredths. Tissue against air is a difference of about 1/3. Now, whenever light crosses a boundary between two materials with different refractive indices, some fraction of it reflects. There is a standard piece of optics called the Fresnel equation that tells you how much. And for light arriving straight on at a flat surface, it is simple enough to do in your head if your head is having a good day. You take the difference between the two indices, divide it by their sum, and square the result. Run it for tissue meeting air. The difference is about 0.35. The sum is about 2.35. The ratio is roughly 0.15. And squaring, that gives you about 2.2%. 2% of the light bounces off at every single surface. Run it for tissue meeting seawater. Difference of about 0.02, some of about 2.69, and the squared ratio comes out near 5,000ths of 1%. Divide one by the other, and you get something close to 400 times. That is the entire reason the open ocean is full of invisible animals and the forest is not. A body in the water is optically almost the same substance as the water. A body in the air is optically a completely different substance from the air, and it announces that difference at every boundary over and over, thousands of times, as the light works its way through. I find this satisfying in a way that is hard to explain because it is one of those cases where a fact that looks biological turns out to be pure physics wearing a biology costume. Nobody in the ocean is better at being invisible than anybody on land. The ocean is just an easier room to be invisible in. Same animals, same tissues, same chemistry, and the medium does most of the work. Which is not to say land has produced nothing. It has produced a handful of serious attempts, and they are instructive precisely because of how much extra machinery they need. Glasswing butterflies have wings you can read through. To get there, they cannot just thin the wing out because the 2% problem does not care how thin your wing is. It cares about surfaces. So the wing membrane is covered in a forest of tiny pillars, irregular in height and spacing, small enough that light does not resolve them as a surface at all. What the light meets instead is a gradual ramp of refractive index, from air up to chitin, spread out over a few hundred nanometers. No sharp boundary, no strong reflection. That is a real solution to the problem, and it required nanostructure. Water does not require nanostructure. Water just requires being mostly water. Glass frogs are the other famous land case, and they are more interesting still, but I want to save them, because they solve a problem that is going to come up later in a much more pointed form. There is a second reason the ocean favors this trick, and it has nothing to do with indices. It has to do with the background. Think about what invisibility actually means. It does not mean emitting no light. It means matching whatever is behind you. A chameleon on a branch has to match a branch. Move it to a leaf, and it has to match a leaf. Move it to a rock, and the whole system has to run again, and the animal needs the sensing, the pigment cells, the control, the metabolic budget, all of it, just to keep up with a background that changes every time it takes a step. Now consider the background in the open ocean at, say, 400 feet down in the middle of nowhere. Look up, and you get a broad, diffuse, blue-green glow. Look sideways, and you get a dimmer version of the same. Look down, and you get the same color again, fading toward black. That is it. That is the background, everywhere, all the time, in every direction, for thousands of miles. An animal that is transparent has skipped the matching problem entirely. It lets the background pass straight through, which means it matches every background at once, and requires no updating whatsoever. Turn the animal around, it still works. Move it up 200 feet, it still works. Change the time of day, still works. Transparency is the only camouflage that does not need to know where it is. That is a big deal for an animal with almost no brain and no energy to spare. Salps and comb jellies are not running a rendering engine. They do not have to. Their concealment is a material property, not a behavior, and material properties do not get tired. So the pelagic ocean sets up a situation where the physics is 400 times more forgiving, and the background problem effectively disappears. Under those conditions, transparency stops being an exotic achievement and starts being something close to an obvious move. And the evidence for that is how many completely unrelated animals have made it. That word, unrelated, is doing heavy lifting there, and it deserves a moment. When a trait shows up once, it is a curiosity. When it shows up in two lineages, it might be inheritance. When it shows up in animals whose common ancestor was a blob of cells in the Precambrian, and each of them arrived at it separately, using different tissues and different tricks, then you are not looking at a family resemblance. You are looking at a solution being found repeatedly because the problem keeps posing itself in the same terms. Comb jellies and glass squid are separated by well over 500 million years of independent evolution. Their last shared ancestor was some early animal about which we know almost nothing, and everything that makes a squid, and a comb jelly, was built after that point in parallel, with no communication between the two projects. And yet both of them are see-through. And both of them are see-through for the same reason. And both of them fail in the same two places. That last part is the part I keep circling back to. Different starting bodies, different construction methods, radically different ancestry, and the failures line up, which suggests the failures belong to the requirement itself and never were accidents of construction. So how do you actually build a body that light ignores? There turn out to be about 4 routes, and most transparent animals use some combination of them instead of picking one. It is worth walking through each, because the choice an animal makes here determines what kind of animal it can be, how big it can get, and how fast it can move. Route 1 is the brute force approach. Be water. If the problem is that your tissue has a different refractive index from seawater, then the crudest fix is to make your tissue mostly seawater. Dilute yourself until the difference stops mattering. This is the jellyfish strategy, and the salp strategy, and the comb jelly strategy, and it works spectacularly well. A salp's tunic, the outer casing of the animal, runs over 95% water. There is a study that measured the refractive index of tunicate tunics directly, in an ascidian and in a salp called Thetis vagina, and found the difference between tunic and seawater came out somewhere between 2 thousandths and 15 thousandths at yellow light. That is nothing. That is a rounding error. Light entering that tunic barely registers that it has entered anything. Leptocephaly, those eel larvae I mentioned, run the same play with a twist. Their bodies are built around a jelly-like matrix loaded with glycosaminoglycans, long sugar chains that hold enormous amounts of water in place. And the water content of that matrix comes in around 92%, which makes a leptocephalus something closer to a sheet of structured water with a very thin fish laid over the top. A spinal cord, a gut tube, a few muscle blocks, and 2 eyes. That matrix is doing double duty, incidentally, which is the sort of detail that makes me trust the explanation more. The glycosaminoglycans are not just optical filler. They also serve as buoyancy and as an energy store. And when the larva finally metamorphoses into a young eel, it burns through that matrix and shrinks. The transparency is free in the sense that the animal was going to carry that material anyway for other reasons. Route 2 is subtler, and it is the one that keeps showing up in the animals that actually have to move fast. Instead of removing the structures that scatter light, you organize them. Here is why that works, and it is one of the prettiest results in optics. Scattering from a bunch of tiny particles is not simply additive. Light waves that bounce off 2 neighboring particles can arrive somewhere out of step with each other and cancel out. If the particles are arranged at random, the cancellation is partial and messy, and you end up with a lot of stray light going sideways. If the particles are arranged in a regular lattice, with spacing much smaller than the wavelength of the light, then almost all the sideways scattering cancels itself out, and the only direction that survives is straight ahead. You are carrying an example of this in your face right now. Your cornea is a stack of collagen fibrils. Collagen has a refractive index noticeably higher than the fluid around it, and there is a lot of collagen in there. So by the naive account, your cornea should be about as clear as a fingernail. It is clear instead because those fibrils are laid down in a lattice with remarkably uniform diameter and spacing, all of it well under the wavelength of visible light, and the lattice cancels the scatter. Damage that ordering and the cornea goes cloudy within hours. Corneal edema is exactly that. Water gets in, the spacing goes irregular, and a tissue that has been transparent your entire life turns into ground glass. That trick is available to muscle too, and it has to be. because a transparent animal that needs to swim cannot dilute its muscle to 95% water and still get anywhere. Larval fish and transparent shrimp have muscle you can see through, and the reason is that muscle is already one of the most geometrically ordered tissues in the body. Actin and myosin filaments sit in a hexagonal array with spacing in the tens of nanometers. It is a photonic accident waiting to happen. Turn down the pigment, thin out the mitochondria, keep the array clean, and muscle becomes a legitimate window. Route 3 is the surface treatment. And this is where things get strange. Even after you have matched your interior to seawater, you still have an outer boundary. And that boundary still reflects a little. Only a tiny fraction given the numbers we ran earlier, but in an environment where a predator is scanning for the faintest flicker against a uniform blue field, a tiny fraction can be the difference between eaten and not eaten. Nature's answer, over and over, is anti-reflective nanostructure. Same principle as the coating on a camera lens, arrived at independently by animals, a very long time before we thought of it. That SALP study I mentioned found the tunic's surface covered in nipple-shaped bumps roughly 80 nanometers high, and their optical modeling supported an anti-reflection effect from that array. Small bumps, closely packed, creating a gradual transition in refractive index instead of a sharp step. The best documented case, though, comes from a group of animals called hypereid amphipods, and specifically from a genus called Cystosoma. In 2016, Laura Baggy, Karen Osborne, and Sonka Johnson ran electron microscopy across the cuticle of 7 hypereid species, and they found 2 things nobody had described before. On the legs of Cystosoma, 90 to 100% of the surface was covered by an ordered, repeating array of nanoscale bumps, about 200 nanometers tall and spaced about 96 nanometers apart. Textbook anti-reflective structure at the correct scale in a living animal. The second finding was better. Across Cystosoma and 6 other hypereid species, the body surface carried a continuous single layer of tiny spheres, roughly 52 nanometers across on Cystosoma, packed evenly like a coat of microscopic ball bearings. And the evidence suggests those spheres are not made by the amphipod at all. They appear to be living bacteria. The animal appears to be farming its own anti-reflective coating, or at minimum tolerating it, and the coating cuts reflection by as much as 250-fold. Think about what that means for a moment. An amphipod drifting in the dark at a few hundred fathoms, wearing a fitted layer of bacteria whose job, whether they know it or not, is to keep their host from throwing back the searchlight of a passing lanternfish. That is not a partnership anyone would have proposed on paper. Route 4 is the one that gets least attention and might be the most important. Just do not be very thick. Scattering compounds with distance, every millimeter of tissue the light passes through is another chance to get deflected, and the losses multiply rather than add. Double the tissue, and you more than double the problem, which is why so many transparent animals are shaped the way they are. Flat, ribbon-like, filmy. Drawn out into sheets and threads, a leptocephalus is shaped like a willow leaf, wide and long and almost nothing through the middle. Comb jellies are thin-walled bags. Larvaceans build enormous mucus houses around bodies that are themselves slight. There is a hard ceiling implied here, and it will matter later. Transparency and size pull against each other. The bigger you get, the more tissue light has to cross, and past a certain point, no amount of index matching saves you, which is one reason there is no transparent tuna. There is a version of this same physics running in laboratories right now, and it makes a useful reality check on everything I have just described. For about the last 15 years, neuroscientists have been doing something called optical clearing. The goal is to look at an intact brain, a whole mouse brain, without slicing it into 2,000 sections and reassembling the picture afterward. The obstacle is that brain tissue is white and opaque because it is packed with membranes and fat and protein, all with refractive indices that disagree with the water between them. The solution is not to remove anything at first. The solution is to soak the tissue in a fluid whose refractive index is high enough to match the structures inside it. Push the surrounding medium up from 1.33 toward 1.45, and suddenly the mismatch that was scattering all that light collapses toward 0. The brain does not change. Not chemically, not structurally. It just stops disagreeing with its surroundings, and it turns clear enough to image straight through. There are photographs of this. A mouse brain sitting in a dish over printed text, and you can read the text. It looks like a magic trick, and it is not. It is arithmetic on a refractive index. I bring it up because it settles a question that would otherwise nag at this whole subject. Is transparency about what the animal is made of, or about how it relates to what surrounds it? The clearing experiments answer it flatly. The same tissue can be opaque or clear, depending only on the medium. Transparency is a relationship, not a substance, which is the reason the ocean gets it for nearly free and why the number 400 did so much work a few minutes ago. All right, time to go through the actual animals, because the abstract version only takes you so far. And the interesting part is watching different lineages solve the same brief with completely different equipment. Start with the ones that are the closest thing to a pure case. Salps look like small, transparent barrels drifting in a chain. And if you have seen one on a beach after a bloom, it probably looked like a used contact lens. Underwater, alive, in a chain of 40 or 50 individuals, they are one of the more surreal things in the ocean. They pump water through their own bodies to move, filtering food out on the way, so their swimming and their eating are the same action. They are also tunicates, which means, awkwardly, for our sense of hierarchy, they are more closely related to you than a lobster is. They belong to our own broad branch of the animal kingdom. They just went a very different direction with it. A salp is close to the theoretical ideal because it has almost nothing to hide. Very little pigment, very little dense tissue, a body plan built around a water channel. The tunic surrounding it is over 95% water with the anti-reflective bumps on the outside. What is left that could possibly be visible? The gut and a nerve ganglion. And that is essentially the list. If you look at a live salp in the water, that is exactly what you see, a faint outline and one small dark or orange lump about a third of the way along, sitting there like a smudge on an otherwise clean window. That lump is the digestive organ, sometimes called the nucleus, and it is the entire visible signature of the animal. I want to flag that because it is the first appearance of the pattern this whole case is built on, and it shows up in the simplest transparent animal available. Even here, in a creature that is barely more than an organized volume of seawater, the thing you can see is the gut. Larvaceans go further and stranger. These are tiny tunicates, distant cousins of the salps, and they build themselves houses out of mucus. Not a shelter exactly, more like a filtration apparatus, a balloon of secreted material with intake channels and fine mesh filters that strain particles out of the water while the animal sits inside beating its tail. When the filters clog, and they clog fast, sometimes within hours, the Larvacean abandons the house and builds a new one. Those discarded houses sink, and there are researchers who argue they are a major route by which carbon gets out of the surface ocean and down into the deep, which is a strange amount of planetary importance for an animal you could lose in a teaspoon. The giant ones, in the genus Bathochordaeus, reach a few inches in body length and build houses over 3 feet across. And the animal inside is highly transparent, which is doing something clever, because the house itself is nearly transparent too. What a predator is confronted with is a faint cloud of mucus containing a faint animal. The signal is smeared across a large volume and diluted at every step. Comb jellies, the ctenophores, come at it from a completely different ancestry and land somewhere similar. They are gelatinous, thin-walled and mostly water, and they move using 8 rows of fused cilia that beat in waves. Those comb rows diffract light as they beat, which is why photographs of ctenophores show these running bands of rainbow. No bioluminescence involved, and no pigment either. It is a diffraction effect, the same reason a compact disc throws colors, which raises an obvious objection. If you are trying to be invisible, why are you wearing a moving rainbow? The honest answer is that, at depth, in dim blue water, there is not enough light and not enough spectrum for the effect to be conspicuous. Those spectacular photographs are taken with bright artificial lights at close range. In the animal's actual optical environment, the comb rows are subtle. It is a good reminder that a lot of what we find striking about deep-sea animals is an artifact of us pointing a floodlight at something that has never in its evolutionary history encountered a floodlight. Then there are the glass squid, family Cranchidae, and these are the ones I would put forward if I had to make the case in a single animal. They are cephalopods, full cephalopods, with the arms and the beak and the brain and the big camera eyes. And a number of them, particularly as juveniles, are transparent enough that you can see clean through the mantle to the water on the other side. They solve the density problem with chemistry. A glass squid carries a large fluid-filled cavity, and that fluid is loaded with ammonium ions, which are lighter than the seawater they displace. It gives the animal neutral buoyancy without a swim bladder and without constant swimming. It also means a large fraction of the animal's volume is a bag of clear liquid, which is optically about as good as it gets. What is left after that is the beak, which is hard and dark, the eyes, and the digestive gland. And what those squid do about the eyes and the digestive gland is, I think, the single best illustration of the argument I am building here. But it is going to make more sense once the two problems have been laid out properly, so park the squid for the moment. The vertebrates got there too, which surprises people because we tend to assume a backbone rules it out. It does not rule it out. It just puts a clock on it. Go out at night over deep water with a light and a fine net and pull up whatever is drifting near the surface, and a large share of what you get will be larval fish, and a large share of those will be transparent, not translucent. Transparent, with a visible spine and 2 black dots for eyes, and a little sausage of gut, floating in a body you can barely resolve against the water in the jar. This is close to universal for fish that spawn into open water. The eggs drift, they hatch into something a few millimeters long with no speed, no armor, no teeth, and no hiding place. And the one defense available at that size is not being detected. So they are transparent. Almost all of them. Then they grow, and they lose it. Scales come in, pigment cells spread across the skin, muscle thickens, the blood fills with hemoglobin, the body gets deeper through the middle. Transparency drains out of them over a few weeks, and the fish that emerges looks like a fish. I think that developmental sequence is more revealing than any single adult species, because it shows the trade-off happening inside one lifetime instead of across a phylogeny. Transparency is what you use when you are small and defenseless. The moment you can afford muscle and speed and a real skeleton, you stop paying for it, because it is not free and the alternative works better. The leptocephali stretch that window further than anyone. Eels, tarpon, bonefish, and their relatives all go through this stage, and it can last months. In some species, it may run past a year, though the estimates there are soft and depend on reading growth marks, whose timing is not fully pinned down, so treat the long numbers with some caution. What is solid is that these larvae are drifting in open water, in plain view, for a very long time, with essentially no defense but their own optical properties. Some species reach a foot in length while still in that transparent ribbon form, which is a remarkable thing for a nearly invisible animal to get away with. And then there are the crustaceans, which is where transparency gets pushed hardest, and where the failures are most exposed. Because a crustacean cannot simply be a bag of jelly. It has a cuticle. It has real muscle. It has to move. Hypereid amphipods are the group to watch. They live in open water, most of them are small, and many are so clear that the standard problem in studying them is finding them at all. Karen Osborne, who works on these animals at the Smithsonian, has described the experience of sorting through a net sample and realizing that the empty-looking water in the tray is not empty. Cystosoma is the extreme case. Some species reach around 6 inches long, which for a transparent animal is enormous, right up against that thickness ceiling I mentioned. And a cystosoma in the water is very close to a ghost. It has the anti-reflective nanostructure on its legs. It has the bacterial monolayer over the surface. Its cuticle is thin and clear. Its muscle is ordered and unpigmented. And when you look at one in a photograph, what you see is a faint outline and one solid, unavoidable, brownish-red thing filling the front of the head. The retina. That is the whole animal's optical failure sitting right there in the front. A body 6 inches long, invisible from end to end, with one patch of pigment that cannot be removed, because removing it would mean the animal was blind. Now, cystosoma does something about it, and I will come back to what, because it is one of the best answers on record. But the shape of the problem is now fully visible, and it is time to state it properly. Here is where the case turns. All of it so far has been solvable. Pigment can be dropped, water content can go up, structures can be ordered, surfaces can be coated, thickness can be reduced. Every one of those has been achieved repeatedly by animals that had nothing in common except the water they were floating in. Two things have not been solved, not partially solved, not solved in some lineages and not others, not solved anywhere by anyone in the entire history of the trait. An eye cannot be transparent, and a full stomach cannot be transparent. Take the eye first because it is the cleaner argument, and because the impossibility there has nothing to do with engineering. It sits in the definition of the organ. What does an eye do? Strip it back. An eye takes light arriving from many directions, sorts that light by the direction it came from, and converts it into a signal. Two operations: sorting, then converting. Both of them are, by their nature, incompatible with letting light pass through undisturbed. Start with converting. A photoreceptor detects light by absorbing it. That is the only way it can work. A photon has to be caught and destroyed, its energy handed to a molecule that changes shape and starts a cascade. If the photon passes through, nothing is detected. Nothing at all. Detection and transmission are opposites at the level of a single particle of light. So the back of an eye has to be a good absorber. It has to be, in a word, dark. The visual pigments themselves absorb, and behind them in most eyes there is a screening pigment layer whose entire job is to catch anything that got past, so that stray light does not bounce around inside the eye and wash out the image. That layer is the black spot you see in a transparent larva. It is the working surface of the organ. Take it away, and you have taken away the organ. And there is no route around it. You cannot make a transparent detector because a detector that lets light through has not detected it. This is not like pigment in skin, where the pigment serves a purpose that might be achieved another way, or dropped entirely if the cost gets too high. Here the pigment is the function. That alone would be enough. But eyes have a second, independent problem, and it is the one that seals the situation shut. Go back to the sorting operation. Light arrives at your face from every direction at once. Before any of it can be useful, it has to be separated out by where it came from, so that light from the left lands on one patch of receptors and light from the right lands on another. Otherwise, you have a light meter. You know how bright it is and nothing else. The way almost every eye on Earth does that sorting is with a lens. And a lens works by one mechanism only. It bends light. It takes rays coming in at an angle and redirects them so they converge on a point. Now recall what makes light bend. Light changes direction when it crosses a boundary between materials with different refractive indices. That is refraction. It is the same effect that makes a straw look broken in a glass of water. No difference in refractive index, no bending. None. Match a lens perfectly to the water around it, and you have a weak lens, a blurry lens, a lens with a soft focus. You have a piece of water shaped like a lens, doing nothing, which puts the transparent animal in a bind that has no give in it at all. Being invisible requires that every part of your body have the same refractive index as seawater. Seeing requires that one part of your body have a refractive index sharply different from seawater. Those two sentences are the same sentence with the sign flipped. The animal must choose. And the choice is not close, because being blind in the open ocean is worse than being visible. It gets more pointed when you look at how strong an underwater lens has to be. An eye in air gets help. Light entering your eye does most of its bending at the front surface of the cornea, where it crosses from air into tissue, and that boundary is a big jump from about 1 to about 1.37. Your cornea does roughly 2/3 of the focusing in your eye. The lens inside is doing fine adjustment. Put that same eye underwater, and the cornea stops working almost entirely, because now there is water on both sides of it, and the indices nearly match. This is why you cannot see clearly with your eyes open in a swimming pool, and why a dive mask fixes it instantly. All the mask does is hand your cornea back its air interface. So a fish, a squid, an amphipod, anything that evolved to see in water, has to do all of its focusing with the lens alone. There is no cornea contribution to lean on. And to get enough focusing power out of a lens surrounded by water, that lens has to be dense, very dense. Fish lenses are nearly spherical for exactly that reason, and they are packed with proteins called crystallins at concentrations that would be pathological anywhere else in the body. The core of a fish lens can reach a refractive index somewhere around 1.5. 1.5, sitting inside a body that is trying to hold 1.34. That lens is not going to disappear. It cannot. It is the densest optical component in the animal by design, and every bit of that density is refracting the light that hits it. Even if the lens itself is perfectly clear, even if it absorbs nothing, it still acts as a lens on the background. It gathers the light coming from behind the animal and throws it somewhere, which produces a bright point, or a dark ring, or a distortion in whatever should have been a smooth blue field. You can see this yourself with a marble in a glass of water. The marble might be flawless glass, absorbing nothing, and you can still find it instantly because it is warping everything behind it. There is a nice symmetry in the failure when you lay it out. The lens is visible because it must refract. The retina is visible because it must absorb. Refraction and absorption were the two ways of being seen we started with, and an eye is required to do both of them well. An eye is, in the most literal sense available, a device built out of exactly the two properties that transparency has to eliminate. That is why there is no transparent eye anywhere in the ocean, and why every list of transparent animals comes with the same footnote. Nearly invisible except for the eyes. See-through apart from two dark spots, clear body, visible eyes. The phrasing repeats across a century of descriptions, written by people who had never read each other, because they were all looking at the same unavoidable thing. So the animals do not solve it. They negotiate with it. And the negotiations are extraordinary, which we will get to. First, the second failure, which is messier and in some ways more revealing. Because it lives in the animal's behavior instead of in a fixed piece of anatomy. You have to eat. And the thing you eat is another organism, which means it has a body, and that body has all the pigment and structure and density that a body has. A copepod is not transparent everywhere. A diatom has a silica shell and chloroplasts. A small fish has muscle and eyes and blood. You can spend 300 million years refining your own tissue until light passes through it without noticing. And then, in one afternoon, you swallow something and become visible from 30 feet away. The shape of this one differs from the eye problem in a way that matters. The eye problem is permanent and constant. The eye is always there, always dark, always refracting. The gut problem is intermittent. Right after a meal, an otherwise invisible animal is carrying an opaque mass around inside itself. As digestion proceeds, that mass breaks down, the structures dissolve, the pigments get processed or excreted, and the animal fades back toward invisibility. Then it eats again. So a transparent animal in open water is oscillating. It is at its most conspicuous immediately after feeding, which is also, unhelpfully, the period when it is heaviest, slowest, and least able to escape. That timing is a rough piece of luck, and it compounds. The moments when the animal most needs to be invisible are the moments when it least can be. Predators do not have to search for a transparent animal at random. They can search for the signature of a transparent animal that just ate, and that signature is more visible, less mobile, and more nutritious than average. Anyone who has watched a small transparent shrimp in a tank has seen this. The animal is a rumor. Then it feeds, and there is a green or orange comma hanging in the middle of nothing, moving around the tank on its own. The gut problem also has layers, and they stack in an order that depends on how deep you are. Near the surface, in water that is still bright, the issue is straightforward silhouette. Food in the gut absorbs light. The gut reads as a dark blob against downwelling brightness, and any predator hunting upward sees a small dark comma traveling through a lit field. That is the version everyone can picture. Go deeper, past 600 feet or so, into what people call the twilight zone, and the light changes character. It gets dimmer, it gets narrower in wavelength until it is basically one shade of blue, and it becomes strongly directional, coming down from above in a shaft with very little scattered sideways light to fill in the gaps. In that regime, silhouettes get sharper, and small contrasts that would vanish at the surface become detectable. Predators down there have huge upward-pointing eyes built specifically to catch shapes against the last of the daylight, so the gut problem gets worse with depth up to a point. And then, below about 3,000 feet, sunlight effectively runs out. Total darkness, in terms of anything the sun sent down. At which point you might think the gut problem disappears, because in the dark nothing is visible, and there is nothing to hide. That would be true if the animals down there stayed dark. They do not. The deep ocean is the largest bioluminescent habitat on the planet, and a substantial majority of the animals living in the midwater column can make their own light. Estimates from video surveys off California put the fraction of luminous animals in the water column at something like 3/4, which is a figure worth treating as approximate given how hard those surveys are to do. But the direction of it is not in dispute. Most things down there glow, which introduces a problem that is, to me, the most beautiful and awful twist in this whole subject. If you are a transparent animal in the deep, and you eat something that glows, you become a lantern. Not metaphorically, physically. The prey is in your stomach, it is still alive or recently alive, its light-producing chemistry is still functional, and many bioluminescent animals flash specifically when they are attacked or disturbed. Being swallowed counts as disturbance, so there is a copepod or a shrimp inside your transparent gut, actively producing light, and your body has spent its entire evolutionary history perfecting the ability to let light pass through it. You have built a lamp housing. You have optimized it for output. There is a real hypothesis in the literature about why some deep-sea animals flash when attacked, sometimes called the burglar alarm. The idea is that a small animal being eaten cannot fight back, but it can make a bright display, and a bright display attracts larger predators to the spot. The larger predator eats the thing that was eating you. That does not help the individual much, but if the display sometimes interrupts the attack, it can pay off often enough to persist. That is an interpretation with supporting observations rather than a closed case, so hold it at that level. But it means the light in your stomach is not a passive accident. It may be an alarm being pulled on purpose by the meal against the diner. So the requirement for the gut turns out to be double, and the two halves point in opposite directions. Above, in the light, the gut must not block light coming through. Below, in the dark, the gut must not let light out. One organ, two contradictory optical specifications, and it depends on the depth the animal happens to be at, which for a lot of these species changes every single day, because vertical migration is the norm in open water. Enormous numbers of small animals rise toward the surface at dusk to feed and sink back down at dawn to hide. So the same gut is expected to be transparent at 3,000 feet at noon and inconspicuous at 200 feet at midnight. Now, before we get to the fixes, there is a third organ worth mentioning, because it demonstrates that the problem is general rather than being about eyes and stomachs specifically: blood. Hemoglobin is one of the most strongly absorbing molecules an animal makes. It has to be. It works by binding oxygen at an iron center, and that same chemistry gives it a fierce appetite for certain wavelengths, which is why it is red. A body full of hemoglobin is a body full of a very effective light absorber, distributed through every tissue in a branching network. Marine transparent animals have mostly dealt with this by not having much blood in the first place. Jellies and salps and comb jellies have no closed circulatory system to speak of. Larval fish and leptocephali simply run at very low hemoglobin during the transparent stage, which they can afford because they are small enough that diffusion does a lot of the work. The Antarctic ice fishes went all the way and lost hemoglobin altogether, which is a fascinating case, though those fish are not transparent for other reasons, so it does not quite belong here. The animal that confronted the blood problem head-on lives in a forest, which is a slightly awkward place for this story to go, and I'm going there anyway, because it is the same physics. Glass frogs live in Central and South American forests. They sleep during the day on the underside of leaves, and their undersides are transparent, so that a predator looking up sees a green leaf with a slightly blurred patch on it instead of a frog. The transparency is real. You can see the heart beating and the gut through the skin. But a frog is a vertebrate, with a full circulatory system and a normal amount of blood, and that blood should ruin the effect completely. A body that is transparent, except that it is full of red liquid, is not transparent. In 2022, a team including Carlos Taboada and Jesse Delia, working with researchers at Duke University and the American Museum of Natural History, figured out what these frogs are doing, and the answer is nearly hard to believe. When a glass frog goes to sleep, it takes roughly 89% of its red blood cells out of circulation and packs them into its liver. Almost 90% of the animal's red blood cells, removed from the bloodstream and stored in one organ every day, and then released again when the frog wakes up and needs to move. Doing that raises the frog's transparency by a factor of 2 to 3. They worked this out using photoacoustic imaging, which uses light to make red blood cells produce sound, so you can map exactly where the cells are inside a living animal without cutting it open or injecting it with anything. The frogs were just asleep in the imaging setup, which is the part I like. No surgery, no dye, no dead specimens. The animal naps, and the machine watches its blood move. And the liver they pack the cells into has a reflective coating on it, so the packed up blood sits behind a mirror instead of showing through as a dark red lump, which is a preview of coming attractions because that mirror trick is exactly what the glass squid does with its stomach. So the two failures are established, and they are permanent. No lineage has fixed them. Nothing is going to fix them because the physics that makes them failures is the same physics that makes the organs work. What the animals have instead is a catalog of workarounds, and this is the part of the subject I find most worth the time, because a workaround tells you more about the constraint than a solution would. A solution closes the problem, and you stop learning from it. A workaround has to bend around the exact shape of the thing it cannot remove, and if you look closely enough at the bend, you can read the shape. Start with the eye, and start with the most obvious move available. Make it small. If the retina has to be dark, then have less retina. Shrink the absorbing patch down to the minimum that still delivers usable vision, and accept whatever loss in image quality that costs you. This is why so many transparent animals have eyes that look disproportionately tiny for the body they are attached to. A transparent shrimp 2 inches long with 2 black dots the size of pinheads. A larval fish, where the eyes are a smaller fraction of the head than they will be in the adult. The cost is real, and it is not small. Visual sensitivity depends heavily on how much light you can collect and how many receptors you can devote to the image. Cut the retina down, and you are cutting resolution, sensitivity, or both, in an environment that is already dim. So the animal is trading the ability to see for the ability not to be seen, and it is making that trade knowing, in the only sense evolution knows anything, that being detected is worse. Which is a striking ranking when you say it out loud. These animals would rather have poor vision than be visible. In an ocean full of predators that hunt by sight, the defensive value of invisibility outweighs the offensive value of good eyesight. That tells you something about the balance of danger out there. The second move is to spread the retina out. The absorbing pigment has to be there, but nothing says it has to be there in a compact lump. If you take the same quantity of photoreceptor and smear it across a much larger area as a very thin layer, the total amount of pigment is unchanged, but the visibility of any given patch of it drops, because contrast against the background depends on how much light a given area of your body removes relative to the water beside it. Cystosoma appears to do exactly this. Its eyes cover most of the top of the head, an enormous area for a 6-inch animal, and the light-sensitive layer underneath is remarkably thin, spread out into a broad sheet instead of a concentrated patch. Above that sheet sits a deep, transparent layer of optical elements, crystalline cones, that carry the light down from the surface of the eye to the receptors below. Look at a photograph of a cystosoma, and you can see the result. The head is huge and mostly clear, and the visual apparatus reads as a faint pinkish haze across the top. Nothing like a hard black spot. The pigment is all still in there. It has just been rationed out over enough territory that no part of it is dense enough to punch a hole in the background. Then there is Phronima, which took the same principle and pushed it into geometry that sounds invented. Phronima is a hypereiid amphipod, a small transparent one, and if you have seen it referenced anywhere in popular culture, it was probably as the animal that supposedly inspired the design of a certain movie monster, a claim that gets repeated a lot and does not seem to trace back to anything the film's designers actually said. What Phronima is famous for among biologists is different and better. The female attacks a salp, hollows it out, and lives inside the empty barrel, pushing it through the water and raising her young in it. She turns another animal's body into a nursery. The eyes are the part that matters here. Phronima has 4 of them, 2 pairs, and the arrangement is unlike anything else. The upper pair take up essentially the entire roof of the head. They face straight up toward the surface, toward the shaft of downwelling light where prey silhouettes appear. Those upper eyes have very large facets and a narrow field of view, which is the correct design for detecting a faint shape against a dim uniform background at a distance. And here is the trick. The light collected by those enormous upper eyes does not land on a retina at the surface. It is funneled down through crystalline cones that are more than 20 times longer than those in the lower pair, reaching around 5 millimeters in a large individual, which is a substantial fraction of the animal's total length. Those cones act as light guides, carrying the image down through the transparent head to a small retina sitting about 5 millimeters below the eye's outer surface, tucked deep in the body. Sophronyma has separated the two functions. The part of the eye that has to be big, the collecting aperture, is transparent. The part that has to be dark, the retina, is small and buried. That is a beautiful piece of engineering, and it is worth being clear about what it does and does not achieve. The retina is still opaque. It is still in there. It has not been made invisible. What has been achieved is that the opaque part has been reduced to the smallest possible volume and moved out of the most conspicuous position, while the animal keeps a large light-gathering surface that costs it nothing optically because that surface is clear. The constraint has not been beaten. It has been isolated, minimized, and relocated. That is what all of these workarounds are doing, and it is why I keep calling this a compromise rather than a solution. There is a variation on this in fish larvae that goes even further, and it is one of the odder-looking things in the ocean. There is a family of deep-sea fishes called the barbelled dragonfishes, and the adults are the sort of thing that ends up on posters. Long, black needle teeth, a glowing lure on a chin barbell, the whole nightmare package. One genus, Ideacanthus, includes the black dragonfish. The larvae look nothing like that. They are transparent ribbons, about half an inch to just over an inch long, clear, except for a row of small color spots drifting in the upper water where there is still light. And their eyes are on stalks, not slightly protruding, on stalks, long, thin, flexible cartilaginous rods that carry the eyes out away from the head, and in some individuals, those stalks run to something like a quarter of the animal's total body length. In extreme cases, they have been described at close to half the body length. Picture a clear ribbon of a fish with two small dark beads floating out in front of it on threads. This condition is common enough among certain deep-sea fish larvae that it has its own name. It was described so long ago and looked so unlike any adult fish anyone recognized that early naturalists gave the larvae their own genus, Stylophthalmus, before anyone worked out that these were the juvenile stage of animals already known under other names. That is a nice reminder of how thoroughly metamorphosis can disguise an animal from science. What the stalks are for is not fully settled, and it is worth saying that plainly rather than picking the most satisfying explanation and presenting it as fact. The interpretation that appears most often is that the stalks extend the larva's field of view, letting it scan a wider volume of water for prey and for approaching predators, which matters when you are drifting in open water with no cover. There is a second reading that fits everything discussed so far, and I will flag it as my own interpretation rather than an established finding. In a transparent animal, an eye is a dark object with a recognizable shape and position. What a visual predator is actually doing is not detecting darkness so much as detecting pattern, a configuration that reads as an animal. Two dark spots close together at the front of an elongated shadow is the most recognizable pattern in the ocean, because it is the pattern of a face. Push those spots out on transparent threads and you have taken the pattern apart. The two dark points are now separated out of proportion, floating away from anything that looks like a body, connected by a stalk that is itself invisible. What remains is two small, unrelated specks in the water, and specks are everywhere out there. Marine snow, larvae, debris, fragments. The ocean is full of small, dark things that are not worth chasing. Whatever the primary driver, the anatomy is real, and it is documented. And the other detail that comes with it is telling. As the larva metamorphoses toward the adult, the eye stalks retract into the head, and this happens as the fish moves down into deeper, darker water. The stalks belong to the phase of life spent transparent in lit water. When the animal stops being transparent and stops living in the light, they go away. That is the kind of correlation that makes the transparency reading hard to dismiss entirely, even if it does not prove it. Now for the one I think is the single cleverest answer to the eye problem anyone has found, and it was published only recently. The problem, stated once more, the eye has a patch of absorbing pigment that cannot be removed, and that patch shows up as a dark spot against a lit background. But look at that sentence again. The spot is a problem because it is dark. It is dark because it absorbs the light that hits it and sends none back. What if you put something in front of it that returns light instead? If you could cover the front of the eye pigment with a surface that reflects the same color and roughly the same intensity as the water behind the animal, the eye would stop reading as a hole in the background. It would read as more background. That is what a lot of larval crustaceans are doing, and the material they do it with is remarkable. In a study published in Science in early 2023, a group of researchers found that the eyes of many larval decapod crustaceans, the group that includes shrimp, prawns, crabs, and lobsters, carry an ultra-compact reflector lying over the dark eye pigment. The reflector is built from nanoscale crystals of a compound called isoxanthopterin, a pterin, chemically related to the pigments in butterfly wings and in the eyes of insects. The crystals are formed into tiny spheres, and those spheres are packed together into what physicists call a photonic glass, a disordered but tightly controlled packing where the size of the spheres and the density of the arrangement together decide which wavelengths bounce back, and the animals tune it. By changing the size of the nanospheres and how they are arranged, the reflected color can be shifted across a range from deep blue through to yellow. Larvae from bluer, clearer offshore water were found to have reflectors set toward blue. Larvae from greener, more coastal water had reflectors set toward green. The eye is colored to match the specific water the animal is living in, which means these larvae are not hiding the eye pigment. They are painting over it in a color mixed to match the local ocean, using a nanostructure fine enough to be tuned by adjusting the diameter of individual spheres, and it may be doing a second job at the same time. The researchers noted that a reflector sitting between photoreceptors could also improve the eye's performance, either by bouncing light back through the receptors for a second chance at absorption, which improves sensitivity, or by acting as an optical screen between neighboring receptor units, which improves the sharpness of the image. Both of those are real functions in other animals' eyes, so the structure may be paying for itself twice, once in camouflage and once in vision. If that holds up, it is a rare case where the trade-off actually softens. Almost everything else in this story costs the animal something. There is one more approach to the eye, and it is the most audacious of the lot, because instead of hiding the dark spot, it replaces the missing light directly. Think about what a predator below sees when it looks up at a transparent animal with dark eyes. Blue field, and two small gaps in the blue where the light did not make it through. The eyes are removing photons that should have arrived, so put the photons back, build a light source, aim it downward, and set its brightness to match the daylight that the eye is blocking. Now the predator looking up sees an unbroken blue field because the missing light has been replaced by manufactured light of the same color and intensity. This is called counter-illumination, and it is one of the most common defenses in the twilight zone. Lanternfish do it along their bellies. Hatchetfish do it. Certain small sharks do it. The firefly squid does it. whole bodies covered in downward-pointing light organs, tuned to erase the animal's own shadow. What the glass squid do is the specialized version. The photophores of the family Cranchiidae have been described in detail, and the survey work by Peter Herring and colleagues found examples of light organs on the eyes, on the digestive gland, and on the arm tips across all 13 genera in the family. Light organs on the eyeballs. The animal is transparent almost everywhere. The eyes are the exception, so that is where the lights go, positioned to shine down and cancel out the shadow that the eyes themselves are casting. The lamp is bolted to the exact spot that needs erasing. I find that a very clarifying piece of anatomy because of what it implies about the accounting. Bioluminescence is expensive. You need the chemistry, the tissue, the control system, the energy. An animal does not evolve dedicated light organs on its own eyeballs unless the shadow those eyeballs cast is costing it more than the lights cost, which tells you how sharp the pressure is out there. A dark spot 2 millimeters across in dim blue water was worth building a lamp to erase. There is a second thing the eye photophores may be doing, and this is where the twilight zone gets properly strange. Some of these squid have their eyes arranged asymmetrically, and some sit at depths where the downwelling light is only present in a narrow band. Getting counter-illumination right means matching intensity to the local light level, which changes with depth, with time of day, with cloud cover at the surface hundreds of feet above. Some counter-illuminating animals have sensors that monitor ambient light and adjust their output to match. That is a closed feedback loop, running continuously in an animal with a brain the size of a pea. So, to gather up the eye problem before moving on, the eye cannot be transparent for two independent and equally binding reasons. It must absorb, or it detects nothing. It must refract, or it resolves nothing. And here's the catalog of responses, arrived at by arthropods, by fish, and by cephalopods, 3 phyla that have been evolving apart since before there were animals with jaws, and none of which could have copied one another. Shrink the retina and accept worse vision. Spread the retina into a thin sheet so no patch of it is dense. Separate the collecting optics from the detecting surface. Keep the optics clear and bury the detector deep in the body on a light guide. Push the eyes out on transparent stalks so they stop reading as a face. Coat the pigment with a tuned nanoscale reflector that returns the exact color of the surrounding water. Build a light organ on the eye and manufacture replacement photons. 6 strategies. Every one of them is a way of managing a dark spot that will not go away. Not one of them is a way of removing it. That is the signature of a hard constraint. When a problem gets attacked from 6 unrelated directions, and the same residue survives in all 6 outcomes, you are looking at a wall rather than a shortage of imagination. The gut, now, and the gut, is where the workarounds get more varied, because the gut has more degrees of freedom. An eye has to be at the surface of the body, pointing outward in a fixed location with a specific optical geometry. A stomach can be almost anywhere, any shape, any orientation, and it can be lined with anything. So the responses fan out. Response 1 and the simplest: keep it small and keep it moving. Many transparent animals have unusually rapid gut transit. Food goes in, gets processed fast, and what cannot be used goes out quickly instead of sitting in a holding chamber. Salps are extreme about this. They are continuously filtering, continuously processing, and continuously packaging waste into dense fecal pellets that they release, and those pellets sink fast, which incidentally makes salps important players in moving carbon from the surface ocean to the deep. From the camouflage side, the effect is that the visible payload inside the animal at any one moment is kept small. There is a cost, and it is the obvious one. Fast transit means less complete digestion. You extract less from each meal. An animal doing this is paying for concealment in calories, which is a strange line item, but it is the same line item as everything else in this story. Transparency is never free. It is purchased, and different animals pay in different currencies. Some pay in vision, some pay in speed, some pay in muscle. Salps pay in digestive efficiency. Response 2 is compression and positioning, and the glass squid deserve their moment here. A glass squid is, as established, close to a bag of clear ammonium solution with arms. The mantle is transparent, the fins are transparent, the head is largely transparent, the eyes are handled with photophores. What is left is the digestive gland, the organ that does roughly what a liver does, and which in cephalopods is a substantial piece of tissue that has to be dense, because it is doing serious chemistry. There is no version of that organ that is invisible. It is full of enzymes, storage material, waste products, and the partly broken down remains of whatever the squid last caught. So the squid does two things with it, and both are geometry problems solved correctly. First, the shape. In many cranchids, the digestive gland is drawn out into a long, narrow, cigar-shaped or needle-shaped structure instead of a compact lump. Take a fixed volume of opaque material and stretch it into a needle, and the cross-section you present in any single direction collapses. From the side you see a line. From the end you see a dot. Second, and this is the part that crosses from sensible into clever, the orientation is actively maintained. The most dangerous direction to be seen from, in the twilight zone, is below. That is where the hunters are, looking up, using the last of the daylight as a backlight. And the squid keeps that needle-shaped gland oriented vertically, pointing down along the axis of the incoming light so that a predator looking up at it sees the smallest cross-section available, the end of the needle instead of its length, which requires the squid to hold that organ in a fixed orientation relative to the water surface, while the rest of its body pitches, rolls, swims, hunts, and hangs at whatever angle it likes. The gland stays vertical, while the animal does not. There is a version of the same trick in some transparent shrimp and amphipods, where the gut is kept aligned to minimize the silhouette from below. And there is a related version in fishes that went silver instead of clear, which we will come back to at the end, because silvering and transparency are answers to the same question, and they split at a very specific point. Third, on top of shape and orientation, the covering. In many transparent midwater animals, the visible organs are wrapped in reflective tissue, an actual mirror, built out of stacks of thin crystal platelets, usually guanine, spaced at intervals that reflect light of a particular wavelength efficiently. The same material that makes fish scales flash, applied as a sleeve around an internal organ. Why would a mirror help? It seems like the opposite of camouflage. Mirrors are shiny. Shiny is conspicuous. In the open ocean, it is exactly right. And the reason is that a mirror shows you whatever is beside it. Out there, at a given depth, the light coming from any horizontal direction is essentially the same as the light coming from any other horizontal direction. Uniform, blue, symmetrical, all the way around. So a vertical mirror hanging in that water reflects light from one side toward a viewer on the other side. And because both sides look identical, the mirror reproduces the background exactly. It disappears. That is why so many open ocean fish are silver on the flanks. Herring, sardines, mackerel, tuna. Those flanks are precisely vertical mirrors, and the reflecting platelets are oriented to stay vertical even when the fish tilts, which is a documented refinement in several species. It is camouflage by pure reflection, and it only works in an environment where the light field is horizontally uniform. Try it in a forest, and you have made a disco ball. So an animal with an opaque organ can wrap that organ in a mirrored sheath and let the sheath do the work. The organ still absorbs everything that reaches it. Nothing reaches it, because the mirror sent the light back out before it got there. The glass frog turned out to be doing this too, which is why I mentioned it earlier. The liver those frogs pack their red blood cells into has a reflective coating over it. The blood goes into a container, and the container is mirrored, so a predator looking up through the transparent belly of a sleeping frog does not see a red lump. It sees a reflection. Two animals, an ocean and a rainforest apart, a mollusk and an amphibian, with no shared ancestry for this trait whatsoever, arriving at the same answer to the same problem. Store the opaque material in one place, then put a mirror in front of it. And the fourth response is the one that only makes sense in the dark, and it is my favorite thing in this entire subject. Paint the stomach red. Go back to the lantern problem. A transparent animal in the deep swallows something bioluminescent, and the prey lights up inside it. Mirrors will not fix that, because the light is being generated on the inside. A mirror facing outward keeps external light out, and it also keeps internal light in, bouncing it around, which if anything makes the situation worse by giving the light more chances to find a way out. What you actually need, for the first time in this whole story, is an absorber. You need the inside of the stomach to be black. But here is where the deep sea does something that sounds like a riddle. Down below about 2,000 feet, sunlight is gone. And essentially all the light that exists is bioluminescence. And bioluminescence in the open ocean is overwhelmingly blue. There are exceptions, and they are famous exceptions. But the enormous majority of light-producing animals down there emit in a narrow band of blue-green, roughly matching the wavelength that travels furthest through seawater, which means the only light there is to hide from is blue. And a red surface is, by definition, a surface that absorbs blue. Red pigment is what you get when a material soaks up the shorter wavelengths and returns the longer ones. Shine white light on it and it looks red, because red is what comes back. Shine blue light on it and nothing comes back at all, because it ate the blue and there is no red in the incoming light for it to return. In the deep ocean, red is black. Functionally, optically, completely black, because the only illumination available is the exact color that red pigment destroys, which is why the deep sea is full of red animals that no one down there can see. Shrimp, prawns, jellyfish, siphonophores, amphipods, bright scarlet, deep crimson, wine dark. Bring them to the surface, put them under a deck light, and they are some of the most vividly colored animals on Earth. Leave them where they live and they are invisible. Red is the cheapest black available, and it is cheap because carotenoid pigments are already in the food chain and easy to sequester. Now apply that to the gut. If you are a transparent animal that eats things that glow, you line the part of your body that holds those things with red pigment. The prey lights up, and its blue light hits a wall that absorbs blue completely, and nothing gets out. The clearest example anyone has ever imaged is a comb jelly called Lampoctis cruentiventer. Researchers at the Monterey Bay Aquarium Research Institute described it, and the common name they gave it is the bloody belly comb jelly. The animal is a deep reddish-purple ctenophore, and the stomach in the middle of it is a deep, saturated blood red. Everything around that stomach is doing the standard transparency job. The stomach is doing the opposite job on purpose, because the stomach is where the light is going to come from. There is footage of one where the comb rows are firing rainbows down its flanks, and the belly sits there like a dark red bruise. And once you know what the red is for, it stops looking like decoration and starts looking like a piece of equipment. The same solution turns up in animals with no relation to comb jellies at all. Atolla, a deep-sea jellyfish with a distinctive scalloped bell, is deep red, and its stomach is a particularly intense red. Voragonema pedunculata, another deep medusa, has been described with the same blood-red gut. These are scavengers and predators taking in luminous material regularly. And the interpretation that the red gut is there to contain that light is the standard reading in the field. It is an interpretation supported by the optics and by the pattern of which animals have it, rather than a directly demonstrated mechanism, so I will keep it at that level. But the pattern is hard to explain any other way, and it repeats across lineages that share nothing but the depth they live at. Some deep-sea fishes use black in place of red for the same job, lining the stomach and the body cavity with heavy dark pigment, so that a swallowed lanternfish, still flashing, does not turn the predator into a beacon. Dragonfishes are the ones usually cited. A fish that hunts by luring prey with its own light cannot afford for that prey to keep broadcasting from inside it. And there is a coda to the red story that is too good to leave out, even though it belongs to the predator side. A few deep-sea dragonfishes in the group that includes Malacosteus have light organs below the eyes that produce red light instead of blue. Red light does not travel far in seawater, so it is useless for long-range signaling. But that is the point. Almost nothing down there can see red, because almost nothing down there has evolved receptors for a color that does not exist in their world. So these fish carry a private searchlight. They illuminate the water in front of them in a color their prey cannot detect. And they carry the specialized visual pigments needed to see the returning red light themselves. The red animals that spent all that evolutionary effort becoming invisible to blue light are lit up like a stage. I like that one because it is a reminder that none of these solutions are permanent. Every optical strategy in the ocean is a move in a game where the other side is also moving. And any strategy that works well enough for long enough creates a payoff for whoever figures out how to break it. All right? That is the gut catalog, and it is worth laying it out the way we did the eyes, because the parallel is the point. The gut cannot be transparent because food is opaque and food is what a gut is for. Responses: keep the gut small and push material through fast, at the cost of digestive efficiency. Draw the organ out into a needle so it has almost no cross-section. Hold that needle vertical, actively, regardless of body posture, so the smallest possible profile faces the predators below. Wrap the whole thing in a guanine mirror, so it reflects the water beside it. Bolt photophores onto it and replace the light it blocks. Line it with red or black pigment, so that anything glowing inside stays inside. 6 responses again, from ctenophores, cnidarians, tunicates, crustaceans, cephalopods, and fish. 6 phyla, give or take how you count them. And once again, not one of those responses removes the problem. Every single one of them is a way of packaging an opaque object so that it costs less. So we can now state the finding, which is what the whole case has been assembling toward. Every transparent animal in the ocean is built around 2 objects that will never be invisible. And because those 2 objects cannot be eliminated, they have to be managed. And management leaves marks: the needle-shaped liver, the eyes on threads, the bacterial coat, the red stomach, the lamp on the eyeball, the tiny retina buried 5 millimeters down a crystal light pipe. Every one of those features exists because of a limit, and every one of them is legible. Look at a transparent animal and find the parts you can see. Then look at what has been done to those parts. That is the animal's entire evolutionary negotiation, sitting on the outside where anyone can read it. There is one more movement to this, and it is the question that hangs over the whole subject once you accept that transparency is expensive and imperfect. If it is this hard and this compromised, why does it keep happening? And more usefully, where does it stop? There are 4 boundaries that close in on transparency, and each one of them ends up handing the environment over to a different strategy. Knowing where those boundaries sit explains the distribution of camouflage in the ocean better than almost anything else. The first boundary is size, and it is the least negotiable. Scattering accumulates along the path light takes through a body. Each interface is a small probability of being deflected, and the probabilities compound instead of adding. Double the thickness, and you do considerably worse than double the loss of clarity. In practice, this means transparency degrades fast as an animal gets bigger, and it degrades in the thickest dimension first, which is why the transparent animals are, without exception, small or thin or both. The largest properly transparent things in the ocean are the gelatinous ones, and they get their size by being mostly water and mostly hollow. A siphonophore can stretch to over 100 feet, which sounds like it breaks the rule. And it does not, because a siphonophore is a chain of small units strung along a thread. No part of it is thick. Light crossing it passes through a few millimeters of tissue at most, no matter how long the animal is. Take an animal that needs real muscle mass, a dense skeleton, a large liver, thick blood, and the path length through its body goes to inches. At that point, no amount of index matching helps. You can get the refractive index difference down to 1,000th, and it will still not save you, because you have too many millimeters of tissue for the small residual scattering to stay small. This is the ceiling that ends the transparent phase of every larval fish. The animal grows, the light has further to travel, the clarity fails, and long before it fails completely, the fish has already switched to a different defense. The second boundary is at the top of the water column, and it is ultraviolet. Near the surface, sunlight arrives with a significant ultraviolet component, and ultraviolet damages DNA. Land animals and shallow water animals deal with this by absorbing the UV in an outer layer, in skin, in a shell, in pigment. Melanin is very good at it. A transparent animal has thrown away all of that. Light passes through it, which means ultraviolet passes through it, straight into every cell it contains. There is no shielding layer, because a shielding layer is the exact thing transparency forbids. The workaround is a family of compounds called mycosporin-like amino acids, and they are a real answer with a real elegance to them. They absorb strongly in the ultraviolet, and hardly at all in the visible range. So an animal that loads up on them stays transparent to the light that matters for camouflage while blocking the light that damages it. Copepods carry them. Krill carry them. Most animals pick these compounds up through their diet, tracing back to algae and bacteria, which means an animal's ultraviolet protection can depend on what it has been eating lately. The catch, and this is an interpretation rather than a settled result, is that a body which strongly absorbs ultraviolet is a body which is visible to anything that can see ultraviolet. And plenty of marine animals can. Some fish have ultraviolet-sensitive receptors, and the leading explanation for why is that it makes transparent zooplankton stand out against the water. If that holds, then the sunscreen and the camouflage are pulling against each other, and an animal near the surface has to pick its poison, which is one reason transparency thins out in the very top layer of the ocean and gets more common a bit further down where the ultraviolet has already been filtered out by the water above, but there is still enough visible light for camouflage to matter. The third boundary is the sharpest one, and it is the reason I do not think of transparency as a solved defense at all. Polarization. Light waves vibrate, and they vibrate in a particular orientation. Sunlight scattering through seawater ends up partially polarized, meaning the vibrations line up preferentially along one axis. Human eyes cannot detect this at all. A great many marine animals can, and cephalopods are exceptionally good at it. Now here is the trap, and it is a beautiful one, because the trap is set by the very thing that made transparency possible. Remember root 2, the ordering trick. Transparent tissue works by arranging its structures into regular arrays with spacing below the wavelength of light. Muscle filaments in hexagonal packing. Collagen in a lattice. That ordering is what cancels the scatter. Ordered materials are birefringent, meaning the refractive index they present to light depends on the orientation of that light's vibration. Push polarized light through an ordered biological material, and it comes out with its polarization rotated or split, even if not a single photon was absorbed and not a single one was deflected. So the animal is perfectly transparent in brightness, and it is leaving a signature in polarization. And that signature comes from the internal architecture that transparency required. Nadav Shachar, Roger Hanlon, and Adam Petz published the test in Nature in 1998. They gave squid transparent zooplankton to hunt under two lighting conditions, one with the natural partial polarization present and one without it. Under polarized light, the squid detected their prey at distances about 70% greater, 70% further away, on a target that was, in terms of brightness, equally invisible in both conditions. That is a defense being read straight through, and the animals doing the reading are, in a lot of cases, the same animals that use transparency themselves. A squid can be transparent and can also see through transparency. The arms race is happening inside a single group. The fourth boundary is depth, and it is the one where the whole logic of the strategy inverts. Follow a column of water down and watch the problem change. At the surface, and down through the first few hundred feet, there is plenty of light. It comes from many directions because it has been scattered around by the water and by particles, and the ultraviolet is still present. Transparency works here, but it is under pressure from the ultraviolet. And it is competing with a lot of other options, because there is enough light for color and pattern to function. Between roughly 600 and 3,000 feet, the twilight zone, transparency comes into its own. The light is dim, blue, and directional, dominated by a shaft coming down from above. In that regime, silhouette detection is the main threat, and transparency is close to a perfect defense against silhouette detection, because you cannot cast a silhouette if the light goes through you. This band is where the great majority of transparent animals live, and that distribution is exactly what the physics predicts. Below 3,000 feet, the sun is gone. And here transparency stops making sense. Work through it. There is no downwelling light, so there is no silhouette to cast and nothing to hide against. The only light in that world is bioluminescence, and bioluminescence does not come from above in a broad, even field. It comes in flashes, from arbitrary directions, at close range, often from something that is actively hunting you. Now ask what happens to a transparent body under a flash from the side. Transparency was optimized to pass light straight through. Under a beam from an unexpected angle, some of that light will still scatter, and worse, the animal's internal structures, the lens, the gut, the mirrored organ, the guanine platelets, will catch that beam and throw it back. A transparent body in a searchlight lights up. It glints, it scatters, it shows edges. What you want under a searchlight is the opposite of transparency. You want a surface that absorbs the beam completely and returns nothing. You want black. Or, since the beam is blue, you want red, which is the same thing down there. So there is a crossover point, somewhere in the lower twilight zone, where the optimal camouflage flips from letting light through to eating it. And there are animals that sit right on that crossover and do both. In 2011, Sarah Zielinski and Sonka Johnson published work on two mesopelagic cephalopods, a small octopus called Japetella hyathi, roughly 3 inches across, and a squid called Onychoteuthis banksii, around 5 inches. Both live in the band between about 2,000 and 3,300 feet. Both are transparent by default. Animals were collected from trawls in the Peru-Chile Trench and the Sea of Cortez and tested aboard ship. When the researchers hit them with a flash of blue light, of the kind a bioluminescent predator would produce, the animals turned red, almost instantly. Cephalopods have chromatophores, elastic sacs of pigment in the skin, surrounded by muscles that pull them open, contract the muscles, and the pigment spreads across the skin in a fraction of a second. Relax them, and the sac shrinks back to a dot, and the skin goes clear again. So these animals are running both defenses and switching between them based on what the light is doing. Ambient blue daylight from above, meaning a silhouette threat, stay transparent. Sudden blue beam from an odd angle, meaning a searchlight threat, go red, absorb everything, return nothing. They are reading the optical situation and selecting the appropriate camouflage in real time. And the two camouflages are opposites. That result did more to convince me of the whole framework than any single anatomical detail because it demonstrates the trade-off as an active decision rather than an inference drawn from anatomy. The animal itself is treating transparency as one option among several, valid under some conditions and wrong under others. If transparency were simply the best available answer to being seen, there would be no reason to ever switch off. Below that crossover, in the permanently dark water, transparency essentially disappears from the fauna and is replaced by black and red. And the black gets extreme. In 2020, a team led by Alexander Davis, with Karen Osborne and Sankey Johnson among the authors, reported on what they called ultra-black deep-sea fishes, 16 species, spread across 7 distantly related orders, with skin that reflects less than half of 1% of the light that lands on it. The best of them, an anglerfish in the genus Onerodes, came in around 0.05 of 1%. To put that in the range of things people know about, the blackest butterflies come in somewhere between 0.06 and 0.5 of 1%. And the blackest birds, the ones famous for looking like holes in the world during courtship displays, run from about 0.05 to about 0.3 of 1%. These fish are in the same league, and some of them are better. The mechanism is a continuous layer of tightly packed pigment granules, melanosomes, in the outermost layer of the skin. What makes it work is the packing. In ordinary dark-skinned fish, the pigment cells sit with unpigmented gaps between them, and light that hits a gap can bounce back out. In the ultra-black species, that layer is continuous, with the granules sized and spaced so that light entering the layer gets scattered sideways within it, bouncing between granules, lengthening its path, and giving the melanin more chances to absorb it before it can escape. Scattering, used as a weapon against reflection. The same physical process that ruins transparency, harnessed to make something even darker than plain pigment could manage. And the payoff is measurable. The authors calculated that dropping reflectance to those levels cuts the distance at which a visual predator can spot the fish by more than 6-fold compared with a fish reflecting 2%. More than 6 times closer before you are seen. In an ocean where a predator's search volume grows with the cube of its sighting distance, that is an enormous reduction in the odds of being found. So transparency has competitors, and they carve up the ocean between them along lines that are surprisingly crisp. Silvering is the main rival in the well-lit upper water, and it deserves a closer look because silvering and transparency are attempts at the same trick from opposite directions. Transparency says, let the background through. Silvering says, reproduce the background on your surface. Both produce an animal that matches whatever is behind it, without needing to know what that is. And both depend on the light field being uniform in the horizontal plane, which in the open ocean it is. The difference is what they cost and what they permit. Transparency requires that the entire body be optically neutral all the way through, which constrains what tissues you can have, how thick you can be, how much muscle and blood and bone you can carry. Silvering only requires a surface. Behind the mirror, the animal can be anything it likes. It can be a tuna. It can have a spine, dense red muscle, a full circulatory system, a heart that pushes it through the water at 40 miles an hour. None of that shows because none of it has to. The mirror is a small fraction of an inch thick, and everything behind it is somebody else's problem. Which explains cleanly why the fast open ocean predators are silver, and the drifting small things are clear. It is a straightforward consequence of size and tissue density. Silvering scales. Transparency does not. The mirrors themselves are worth a sentence, because they are not simple. A flat mirror reflects only one wavelength efficiently for a given platelet spacing, so a single spacing mirror would come out colored. Fish get broadband silver by mixing platelet spacings within the same stack, and in several species by tilting the platelets so that they stay vertical relative to gravity even as the fish's body rolls, which keeps the reflection pointed at the horizontal light field where it needs to be. There is also work suggesting that some fish scales are built to reflect without polarizing the light they return, which would defeat exactly the polarization detection that catches transparent animals. If that is right, then silvering has quietly solved a problem that transparency cannot. The other competitor is counterillumination, and it dominates the twilight zone alongside transparency, mostly in animals too dense to be clear. The logic is direct. You cannot stop blocking light, so replace it. Cover your underside in photophores, sense the ambient brightness above you, tune your output to match, and your silhouette closes up. It is expensive. It requires light-producing tissue, an energy supply, sensing, and continuous adjustment. And it fails if you get the intensity wrong, because a belly that is too bright is as conspicuous as one that is too dark. Some animals appear to solve part of this with adjustable filters and lenses over the photophores, but it works at any body size, and that is the crucial advantage. A lanternfish can be a real fish, with muscle and a skeleton and a stomach full of copepods, and still erase its own shadow. Where transparency has a hard ceiling around a few inches of thickness, counterillumination has none. Line those three up, and the ocean makes a kind of sense. Small and thin, in the lit or dim water. Be transparent. Cheapest option, no running cost, works in all directions at once. Large and dense, in the lit upper water. Be silver. Works only in a horizontally uniform light field, but permits any body you like behind it. Large and dense, in the dim water where silhouettes dominate, counterilluminate. Expensive, requires active control, but scales to any size. In the permanent dark, be black or be red, which is the same thing. Absorb everything, return nothing, and hope nobody has invented a red flashlight. That map covers the open ocean. Push past its edges, into shallow coastal water, onto a reef, up a river, and the whole scheme falls apart, which is worth a minute because the reasons are informative. Start with the light field, since that has been the hinge all along. Everything transparency and silvering depend on comes from one property of open water. The illumination is smooth and symmetrical, bright above, dimmer below, identical in every horizontal direction, no edges anywhere. That is what lets a body match its surroundings without knowing anything about them. Now move that body onto a reef. The light is coming off coral in one direction, off sand in another, through a gap overhead in a third, and there are shadows. And the shadows have shapes. A transparent animal in that situation is transmitting whatever happens to be directly behind it, which will differ from what is behind it a moment later, and differ again from what a predator at another angle expects to see there. The trick that worked everywhere in open water now works only from certain viewpoints, and the animal has no way of knowing which viewpoints those are. Worse, on a reef, there is something to hide behind, and hiding behind things is cheap. A crevice costs nothing. An animal that can wedge itself into a hole has no need for optics at all, and every gram it would have spent on transparency can go into armor or muscle or eggs. That is why reef fish are the most spectacularly colored animals in the sea. In a habitat full of structure and full of edges, camouflage is a matching problem with a local solution. And once you are matching locally, you might as well use color. And once you are using color, you can also signal with it. Transparency is a strategy for animals with no furniture. Freshwater is the same story, plus a couple of extra penalties. The physics is fine. Fresh water has almost the same refractive index as seawater, so the 400 to 1 advantage over air is still there. Nothing in the optics prevents a transparent river animal. What prevents it is everything else. Rivers and lakes are shallow, which means a bottom is usually visible, which means there is a background with structure in it. They are frequently loaded with sediment and dissolved organic material, which colors the light brown and makes it strongly directional. There are banks, plants, logs, rocks, all of it giving both predator and prey somewhere to be. And the volume of open, structureless water in a typical lake or river is small enough that no lineage is going to specialize in it for millions of years. The exceptions exist, and they are exactly where you would predict. Glass catfish, in the genus Cryptopterus, live in clear flowing streams in Southeast Asia, holding position in the water column away from cover, and they are transparent enough that you can watch the spine flex and see the swim bladder as a silvery capsule near the front of the fish. Everything opaque in that animal is compressed into a small package behind the head, and the rest of it is a clear blade of muscle with a backbone running down the middle. Same brief, same two visible objects, in a river in Thailand instead of a mile of blue water. The eyes are still there, black and obvious. The organs are still there, compacted and reflective. The strategy did not change because the habitat changed. Only the address changed. There is one more edge to the map that is less intuitive, and it turns the logic upside down. Very murky water reduces the payoff for being transparent. Think about why. In cloudy coastal water, full of suspended particles and plankton and sediment, light gets scattered constantly on its way from an object to an eye. That scattering does two things. It dims the object, and it fills the space between the object and the eye with a haze of stray light, sometimes called veiling light. The result is that contrast collapses over a very short distance. 10 feet of murky harbor water will hide a dark fish about as effectively as 150 feet of clear ocean. So in that environment, the water is already doing the job. A predator's sighting distance is short, no matter what the prey looks like, and an animal that invested heavily in optical camouflage would be paying for a benefit the habitat is handing out for free. Which flips the usual assumption. People tend to imagine that clear water is the safe place and murky water is the dangerous one. And for a small animal being hunted by eyes, it is the reverse. Clear water is where you can be seen from a long way off. Clear water is why transparency is worth the enormous cost of building it. The clearest water on Earth is the open central ocean, far from any coast, where there is almost nothing suspended in it because there are almost no nutrients to grow anything. Those regions are sometimes called ocean deserts, and horizontal visibility down there runs to well over 100 feet in the surface layers. It is beautiful, and it is lethal, and the animals living in it have the most extreme optical camouflage on the planet for exactly that reason. Clarity of water and desperation of prey turn out to track each other closely. There is a fifth entry on that list that I think gets left out of these discussions too often, and it belongs here because it is the most common answer of all. Be somewhere else. Vertical migration is the largest daily movement of biomass on this planet, and it is a camouflage strategy in the loosest sense. Enormous numbers of small animals spend the daylight hours deep in water dark enough that they cannot be seen, then rise hundreds of feet at dusk to feed in the productive surface layer under cover of night, and sink again before dawn. They never solve the problem of being visible in lit water. They arrange to be somewhere else while the water is lit. A lot of transparent animals do this too, which means their transparency is a backup. The primary defense is timing. The optical defense covers the hours when timing is not enough, and the transitions at dusk and dawn when the whole water column is on the move and everything is exposed at once. I mention it because it reframes what transparency is for. It is not the only line of defense for most of these animals, and it does not have to be perfect. It has to reduce the sighting distance of a predator that is hunting during a window the animal has already tried to avoid. Every foot it shaves off that distance is a fractional improvement in the odds, compounded over a lifetime and over a population of billions. That is the honest scale of the benefit. A shorter sighting distance, which shrinks the volume of water a predator can effectively search, which lowers the encounter rate, which over enough generations is the difference between a lineage that persists and one that does not. Now the question the whole thing rests on: why does this keep happening? Not why does it happen once. Once is easy to explain. Why does it happen over and over in lineages that have nothing to do with each other, in animals built from incompatible tissues, so reliably that you can predict it from the habitat before you know anything else about the organism? There are 4 reasons, and they stack. The first is the one we started with, and it does most of the work. The medium is nearly the same optical material as the body. That factor of about 400 in surface reflection does far more than give evolution a small edge to work with. It is the difference between a project that is achievable through ordinary incremental change and one that requires nanostructured coatings before you can even begin. In water, an animal that simply reduces its pigment and raises its water content is already partway there. Each step is beneficial on its own. There is no valley to cross. That matters enormously, because evolution cannot plan. It cannot accept a temporary loss for a later gain. A trait only spreads if every increment along the way is at least as good as what came before. Transparency in water has that property. Transparency in air does not, which is why the land examples all required specialized nanostructure and why there are so few of them. The second reason is that the strategy has no running cost and no control system. Almost every other form of camouflage requires something to be happening. An octopus matching a reef is running sensing, processing, and muscular control continuously. A counter-illuminating lanternfish is manufacturing light, monitoring the ambient level, and adjusting output all night. A cuttlefish changing pattern is spending energy every second it is hiding. A transparent animal is doing nothing. Its concealment is a property of the material it is made from. It works while the animal sleeps, while it is injured, while it is distracted, at any angle, at any depth, facing any direction, against any background. There is no failure mode where the system is switched off. For an animal with a nervous system you could fit on a pinhead and an energy budget measured in fractions of a calorie, that is an overwhelming argument. Camouflage that costs nothing to operate is worth a great deal to something that has nothing to spend. The third reason is that the pelagic realm has no other option available. Take almost any other habitat, and there are alternatives. A reef animal can match a coral. A sand-flat animal can bury itself. A forest animal can be brown and hold still. Those work because there is background structure, and structure gives you something to imitate and something to hide behind. The open water column has nothing. It is the largest habitat on this planet by volume, by an enormous margin, and it is the emptiest. No substrate, no shadows, no texture, no refuge at any scale between the surface and the seafloor. An animal living there and hunted by eyes has a very short list of possibilities, and most entries on that list require either size or energy. If you have neither, transparency is what is left. The fourth reason is the one I think is most underrated, and it explains the word keeps in a way the other 3 do not. Almost every animal in the ocean passes through a small, drifting planktonic phase. Crabs do, lobsters do, sea urchins and starfish do, clams and snails and octopuses do, and a very large fraction of fish do, including the vast majority of species that spawn into open water which is most of them, which means that no matter what a lineage eventually becomes, almost every one of them spends some part of its life as a millimeter-scale animal, drifting in open water with no defenses. The adult can be a crab in a burrow, an urchin wedged in a crevice, a grouper on a reef, a bottom-dwelling flatfish. It does not matter. The larva was out there in the blue, exposed, tiny, and slow. So the selective pressure that produces transparency reaches far past the handful of specialist lineages that spend their whole lives in open water. It reaches, at least briefly, nearly every marine animal that exists. The plankton is a bottleneck that almost all of them have to pass through. And the same optical problem is waiting on the other side of it every single time. That is why transparency keeps reappearing. The problem keeps being posed to everybody, in every generation, in the same terms. And the terms are set by physics, which does not vary from one ocean to the next, or from one century to the next, so the answers do not vary much either. Some lineages pass through the bottleneck and out the other side, and lose the transparency as they grow. Some never leave. Salps and comb jellies and larvaceans have effectively made a permanent home in the conditions that other animals only visit as larvae, and they have optimized accordingly. Glass squid stay transparent as juveniles, and lose it in some species as they mature and move deeper. Leptocephali hold on to it for months. It is the same trait, being expressed for different durations, driven by the same physics, in animals that have almost nothing else in common. And in every one of them, without exception, at every stage where it is expressed, there are two things you can still see, which gives you something you can actually use. And it is the part of this I would want somebody to walk away with. Anytime you see a transparent animal, in a photograph, in an aquarium, in a video from a submersible, you can read its history off it in about 10 seconds. The method is short. Find the parts you can see, there will be two, or a version of two. Then ask what has been done to each of them. Run it on the animals we have been through, a salp. One small dark or amber lump about a third of the way down the body, and nothing else. That lump is the digestive organ. The eyes are absent because a salp has no image-forming eyes to lose, which is why it comes closer to pure transparency than anything with a nervous system worth the name. Cost paid, no vision at all, and a digestive system running fast and inefficient. A larval fish, 2 hard black dots, and a thin sausage. Eyes and gut, undisguised. Because a larva a few weeks old has not had time to evolve anything fancier and will not need it, since the whole phase is temporary. Cost paid. Small eyes, poor vision, a short window of exposure that the animal is trying to grow out of. Cystosoma. A faint pink haze across the top of an enormous head. The retina thinned out and spread wide so that no square millimeter of it is dense enough to punch a hole in the background, with a bacterial anti-reflective coat over the outside of the body. Cost paid. An eye that had to be built at absurd scale to compensate for how thin its light-sensitive layer is. Phronema. A pair of enormous clear windows on the roof of the head, and a small dark speck buried deep in the body, connected to those windows by 5 millimeters of crystal light pipe. Cost paid. A narrow upward field of view from the big eyes, a body plan organized around one direction of vision, and a life spent inside a hollowed-out salp. A glass squid, 2 eyes with lamps under them, and a long needle of digestive gland held vertical, no matter what the rest of the animal is doing. Cost paid. The metabolic bill for bioluminescence, plus whatever postural machinery it takes to keep an internal organ pointed at the center of the earth while you swim. A bloody belly comb jelly. A blood-red stomach in a body that is otherwise doing everything it can to be invisible. Cost paid. A permanently conspicuous organ which is only permissible because down there nothing can see red anyway, and a diet of animals that would otherwise turn the eater into a signal flare. A glass frog, a mirrored liver holding almost 90% of the animal's blood cells while it sleeps. Cost paid, a body that has to nearly suspend its own circulation for hours at a time and then restart it without clotting, every single day. Same reading 7 times. On animals from 4 different phyla. Where is the eye, and what did you do about it? Where is the gut, and what did you do about it? Everything else on those animals is the easy part. I find that a strangely satisfying way to look at biology, and I think it generalizes past this subject. When you can identify the constraint an organism cannot escape, the rest of the organism turns into commentary on that constraint. The interesting features stop being a list of facts and start being a record of decisions. And on the specific question of whether we should be impressed, I have a position, and it is a little contrarian. The impressive part of a transparent animal was never the transparency. Transparency is, as we have covered, close to a physical default for a small, soft body in seawater once the pigment is gone. Take a jellyfish and remove its color, and it is transparent almost automatically. The impressive part is the 2 organs. It is the isoxanthopteran reflector tuned to the local watercolor. It is the needle-shaped liver kept vertical. It is the retina spread into a film. It is the light organ, mounted on an eyeball. Every one of those is a solution to a problem that could not be solved, arrived at by an animal that could not think about it. Which is where I want to put a question to you since you have gotten this far. The thing I keep wondering about is what a transparent animal's world looks like from the inside, from behind those eyes, looking out. Cystosoma has an eye that fills its entire head and a retina too thin to build a detailed picture. Phronima looks straight up through 5 millimeters of crystal, at a shaft of light it can barely resolve. These animals are living in an optical environment with almost no information in it, hunting and avoiding and reproducing on the basis of faint shadows against a blue field. If you want to leave a comment, tell me which of the 6 workarounds you would not have believed if it had turned up in a science fiction novel. My vote goes to the bacteria, an animal wearing a fitted layer of living microorganisms as an anti-reflective coating is the kind of detail that would get an editor's note in the margin saying this is too much. And if this is the kind of thing you want more of, where a question that sounds like a nature fact turns out to be a physics problem with a body wrapped around it, subscribe, because that is essentially the whole channel. Leave a like on the way out if the mechanism landed for you. There is one more thing that ought to be said before this closes, because leaving it out would make the whole subject sound more finished than it is. We do not fully understand how transparent tissue works. That sounds like an odd admission after everything above, so let me be exact about what is known and what is not. The physics is solid. Refractive index, scattering, absorption, the Fresnel relations, the way ordering cancels scatter, the 400 to 1 advantage of water over air. None of that is in doubt. It is standard optics, and it has been standard for a very long time. The optical measurements of whole animals are solid. Johnson and Witter put those numbers on record, and other groups have measured tunics and cuticles and gels since. We know how much light gets through these animals. What sits in between is much shakier. Take a specific transparent tissue, a slab of amphipod muscle, a section of larvation body wall, the mantle of a juvenile glass squid, and ask precisely which structures in it are scattering what, and how the animal has arranged them to keep the scattering low. That question is mostly open. Johnson said as much in his review back in 2001, listing the ultrastructure and optical properties of transparent tissue as one of the main things the field needed to go and look at. And while there has been real progress since, particularly on surfaces and coatings, the interior remains harder. Part of the difficulty is practical and slightly absurd. These animals are extremely hard to collect intact. They are fragile, Many of them are gelatinous enough to disintegrate in a net, and quite a few of them are damaged simply by being brought up through a pressure and temperature change. The species that are easiest to study are the ones tough enough to survive being caught, which is a biased sample of exactly the wrong kind. And then there is the problem of finding them at all. Researchers working on midwater transparency have said, repeatedly, that the limiting factor is that you cannot see what you are trying to collect. The animals are doing their job. They are doing it to us as effectively as they do it to a lanternfish. Remotely operated vehicles have changed this considerably in the last 30 years because they let people watch these animals behaving normally at depth instead of examining wrecked specimens on a deck. A number of the observations in this piece, the vertical digestive gland, the red stomachs, the switching cephalopods, exist because somebody put a camera down there and looked, instead of pulling something up in a net and guessing. So there is more coming on this. Certainly on the tissue side, probably on the eye side, and I would put money on a few more workarounds turning up that nobody has thought of yet because the pattern of the last 20 years has been that every time somebody looks closely at a transparent animal with better instruments, they find another piece of optical engineering that was invisible in both senses. But whatever else turns up, the shape of the answer is not going to change. Because the shape of the answer is set by something that predates all of it. An eye works by absorbing light and by bending it. Those are the two mechanisms by which an object becomes visible. There is no third way to see, and no way around either one. Any animal with a functioning eye is carrying a dark spot and an optical distortion, and it will carry them for as long as it wants to see. A gut works by holding another organism inside a body. That organism was built by the same physics, with the same pigments and the same structures, and it does not stop being opaque when it changes owners. Any animal that eats is carrying, for some fraction of every day, a piece of somebody else. Those are the two lines that transparency has never crossed. Not in ctenophores, which have had 600 million years. Not in the tunicates, who are our own relatives. Not in the crustaceans, who have the finest nanostructural engineering in the ocean. Not in the cephalopods, who can change their entire appearance in a fifth of a second. Every one of them has come up against the same two objects, and every one of them has done the same thing in the end, which is to stop trying to remove them and start trying to manage them. That is what a transparent animal actually is. A body that has surrendered everything it could surrender, and then built a small, ingenious, expensive apparatus around the two things it could not. The needle held vertical, the retina spread to a film, the lamp on the eyeball. The stomach painted the one color that reads as black in the dark. The bacteria on the cuticle. The eyes carried out on threads. Every one of those is a monument to something that could not be done. And if you ever get to see one of these animals in the water, look past the clear part. The clear part is simply what happens when you take enough away. The achievement is the handful of stubborn, unremovable pieces, and what a lineage did with them over hundreds of millions of years of not being able to make them go away. The ocean does not hand out invisibility. It hands out a discount, an enormous one, on the cost of trying. And then it holds back exactly two items, and it holds them back from everybody, in every generation, in every lineage that has ever attempted this. You can see through nearly all of a comb jelly. You will never see through all of it. The limit is the animal.